Hello, everyone. I'm Jun ZOU from China Huazhong Agricultural University. I would like to very worried to have this chance to share some progress of our work for seed protein content in this meeting. My topic is about exploring the value of rich genetic variation for seed protein in a novel gene pool of Brassica napus. I will introduce from this third part. The first is about the construction of a dynamic new type Brassica napus gene pool. And the third part is about the improvement and the trait evaluation for this gene pool. And the third part is to review the genetic basis of seed protein content. The major cultivar rapeseed Brassica napus is a young allo polyploid with limited genetic basis. But in Brassica species, they are rich genetic diversity and annual traits. So it's very achievable. There is a good approach by interspecific crosses to broaden the genetic basis of Brassica napus. So we also use the interspecific crosses with the related species to construct a dynamic new type Brassica napus. Firstly, we constructed developed hex ploids from Rapa and Caminata, and then crossed with the partially new type napus already has the introgression from Caminata and Rapa, and then after selection and subpollinated, got the third generation of the lines with more than 80% of the annual introgression, and then got the inbred population after selection and self-pollinated, and then also at the same time introduced the dominant genic mass sterilized, got the recurrent selection population. By using the selected anid or represented lines of we constructed the bipairing populations. I will introduce these populations later. And on the same time, we further introduced the genetic variation from Jun ZOU to further broaden the genetic diversity, and also introduce the anid variation from Jun ZOU. And this population will also has finished the 10 rounds of recurrent selections. So in the novel gene pool, we will introduce a lot of novel genetic diversity. First is about the pangenome variation across species, the variation among the A, B, and C genome, and the subgenomic variation between the different A and the C genome, and the pangenomic variation within the species. For example, within the Rapa and the Caminata and the Jun ZOU, and also novel variations such as transport activation, homogeneous exchange, and this kind of novel variations induced. And so also by the interspecific crosses and a round of recurrent selection, they will introduce these novel variations. So, and this gene pool, we will found the presented subpopulation level of differentiation with the conventional Brassica napus. And this red part is the new type gene pool and the lines, and they are founder parents, and the blue lines present the conventional napus represented cultivars. So we can find they are formed different subpopulations. And also we can, by the SSM markers and GPS markers, we can find in different loci there are a lot of allelic, multi-allelic variation and novel allelic recombinations. As also, besides of the introgression from the A and the C annual introgression, and there are some minor introgression from the B genome to the A and the C genome. And we also present in this gene pool, we can find the old linkage description block has been broken, and there are a lot of deletions and duplications, and this across the A and C genome. Because the C genome of the Brassica napus present limited genetic diversity, even than the less genetic diversity than the A genome. So from our analysis on the gene pool, we found not only on the C and also on the C genome they present rich genetic diversity, and also introduced a lot of genomic structure variations. The second part is about the improvement on this gene pool by further introduce the Jun ZOU genetic variations. We finished 10 rounds of recurrent selection, and by this selection we can find there from any lines of the interspecific lines, their development traits, seed quantity traits, seed yield traits, they had been improved a lot. And also besides of the rich phenotypic variation from the flowering time, plant height, a lot of traits, there were also specific traits. For example, the glucosinolide from the United Species for introduced to the new type in apples, and also lines with the density of the membrane ports and the port per port seeds, and also there are a lot of good traits. We can find the lines with these traits. And when we crossed with the conventional BNAP testers, they presented a strong heterosis. Not of a recombination we can select it for the breeding cycles with the breeders. For the seed protein content, for this 10 rounds of selection, whatever from the sterilized, also with the sterilized, we found a part of the lines with over 30% with a relatively high frequency, and they are the added part was because we are more focused on the seed oil content. So normally they are 25 averagely % of the protein content, and then in this lines without a lot of very intensive or many years of selection compared to the conventional napus. I think this will be a good resources for the for selection of the seed oil content, seed protein content. So the question is, so how to, so can we use it or to increase the seed protein content, and how to use and select it? Is there a magic cocktail for this trait, or how about the prediction of the on this trait? So we further constructed several biparent populations and also in the diverse populations to invent the genetic basis of the traits. For seed protein content, we also use these populations for the coding mapping and their analysis, or as the training population for further work. So the TNDH population is a training population where already has a lot of genotype and phenotypes. We developed these 3 new populations, then we got a lot of phenotypes and genotypes. For the seed oil, seed protein content in these 4 populations, we can see the seed protein content is also a complex quantitative trait, and by using the phenotypes in different environments, we can identify a lot of cocktails for this. But we can also see a lot of cocktails presented minor effects. We identified a total of 7 cocktails showing exploring the phenotypic variation over 10%, but most of the cocktails showed minor effects. And this cocktail would be further validated, integrated, and compared. We can also to analyze what they are will help to the further genomic prediction models. So since it's a quantitative trait, we think about the genome prediction with this trait. How's the possibility? So this has been, we use the TNDH population because it's the has a lot of phenotype and genotypes, and we evaluate the relationship with the oil content and protein. Is no doubt there are negative correlations, and the heritability of this trait present relatively high heritability. So it is also present it's suitable for the genome prediction in the next step, and the high quality of the data. And we using the phenotype and the genotypes of this training population, we evaluate the effects of mark density, training population size, and number of environments on the prediction accuracy. So this is compared with the oil content. The protein content has a little bit less prediction accuracy, but for the higher prediction, they are over 0.6. It's a relatively high compared to the yellow traits and other. And to improve the accuracy of the genome prediction in a biparent population, we found the recombination events obtained is more important than the density of the markers. And the number of lines has a great impact on the prediction accuracy. But in overall, the larger size, more environment would be also help to increase the prediction accuracy, of course. So the next to improve the prediction accuracy and also implementation this prediction in the breeding cycle, we use the more training populations with the same or similar genetic backgrounds and also the string mapping population, including their derived lines for the test of the new type lines from the novel gene pool and their derived lines. And on the same time, when they are after tested, they will go back to the training populations. So this work is still on the way. So to summarize my presentation today, firstly, we introduced the genetic variation from 4 species to the napus genome, and to develop a dynamic gene pool. And this gene pool would be used to the targets or gene resources for increasing the seed protein content, and also other gene plasmids from in the specific crossings would be also helpful for increasing the seed protein content. And we understand the genetic basis of this trait would be helpful and important for the further for the genetic improvement of this trait. And the genome prediction on protein content would be further implemented with more training population and test populations. And then from the 0.6 to higher prediction accuracy, and also the gene plasm or the breeding lines with different genetic background will have a high accuracy, then would be is a good way to for this trait. So finally, I would like to take this opportunity to thank to the organizing of this GCARC meeting and our national and international collaborators for many years. And I thank the chronicles and the students from our group and our Brassica Research Center of Huazhong Agricultural University, and thanks the funding from the National Natural Science Foundation of China for the support on the GS work on this. So thank you all. Thank you for your attention.